Happy Halloween. So do y'all go trick-or-treating, or is it, younger folk do that? Is a trunk or treat, did that already happen on the weekend? There's some trunk or treat thing going on. Back in the day when my boys were little, the dorms used to be open. You could go into the dorms, and it was awesome because you all had decorated your halls and your room area, like the doorway. Sometimes the entrance to your suite would be decorated. Man, the kids in the community love that. And then COVID, and that all went. So it was really too bad to see that go away. It was, and it seems like the college students also had fun with it. They were laughing and getting into it. So the good old days. Okay, so we have a reminder that the next exam is already next week. Wow. So that means we need to do a review session either late this week or Sunday or Monday. Do a lot of you have night labs on Monday? Would Monday evening work? 5, 6? Any preferences? No. 7? Potentially. Oh, Monday. Now that you said 7, that triggered my memory. I have something going on. I have a night class that I have to deal with that night. Sunday? Sunday? 5, 6 work? Sunday? So let me look at my schedule a little bit more carefully. I'll send out an announcement, and then we'll talk about it toward the end of this week to verify. I'll find a room. If this one's not available, something. So tentatively Sunday, late afternoon or early evening. Of course, for that to be most beneficial for you, you have to be like ready to ask questions. I'm not going to just sit there and lecture to you ad nauseum. So questions on things, and if we have it on Sunday, that would give you enough time to identify your weaknesses and strengths and try to shore up what you need to do before that Friday. Okay. All right, so we're continuing on with hormone as signal transduction players. And so we've thought about hormones already in lab. So some of this should sound vaguely familiar, at least some of the bigger picture ideas. Gibberellins are another group of hormones. So there's many different types of gibberellin-like chemicals that have similar impacts on the plant. Also be aware that it was actually first identified in a fungus that hung out with rice plants. And they named that fungus Gibberella fujikori because it's a plant pathogen that would cause the rice plant to increase its stem dramatically and too quickly, which would then cause the rice plant to flop over. And so they named it foolish seedling disease after the fact that those darn rice plants flopped over. And that's really quite devastating if you're trying to grow rice and then harvest the seeds. Having them flopped over like that is really bad news. So they went into those rice fields and they were investigating why is this happening, and they isolated this fungus, and then it was identified as Gibberella fujikori. So then they coined the term gibberellins, that the hormone that they isolated from this fungus. And then they searched for it in the plant, and certainly they were able to find it there as well. So Often they talk about gibberellic acid as the main hormone in the plant body, but just be aware that plant-associated microbes, good and bad, seem to be capable of producing a gibberellin-like compound. And it is found in a variety of areas of the plant body. We'll talk a little bit more about some of these areas in more detail in a bit, but seeds, germinating seeds, gibberellic acid is important. Young developing leaves, and certainly in the stem tissue, as you saw in lab with the wild type pea versus the dwarf pea, and the impact that the hormone can have. Here's an example of the complication of hormones in plants. You can see that auxins can stimulate gibberellic acid and vice versa. And so these two tend to play with each other, so to speak, and can promote each other, the synthesis of each of those. So again, it's kind of hard to tease apart which hormone is doing what. It's a lot of effort, a very careful experimentation. Just a visual in case you're wondering what it looks like. And so they tend to identify them this way, where it's GA and then a number, subscript number, to refer to the different types of gibberellic acid that are being produced. And so some classic pictures here of, so for example, figure C is showing the dwarf, I think those are cabbage, that wasn't sprayed, and then you can see the ones that were sprayed, how it vastly increased the stem length. Same, pretty dramatic in figure B as well. And so with oh, there it is. I was trying to make sense of that second pot. So you can get my, so here's the dwarf that didn't get sprayed. Here's the dwarf that did get sprayed, and you can see it grew almost as big as the wild type. So very dramatic. This one is a little bit more obscure on the figure A, and no, he's not holding it out. Someone pointed out to me, oh, he's just holding the grapes, the bigger grapes, out closer to the camera. That's why it looks like that. And now, ever since that student said that, that's all I can see when I look at that picture. I never saw that before. But no, he's not doing that. What's going on here is the left grapes were not sprayed, the right ones were. And it's not really the grapes themselves that responded to the gibberellic acid, it's all the stem tissue that the grapes are growing on. What happened was the stem tissue increased in length, which made the grapes space out from each other more. So now they have more physical room to get bigger. And that's what they see happening. So it's pretty common, I believe, that grape producers will spray their grape bunches, and it's not really, they're not really going after the fruit. It's actually the stem. They're trying to get the stem to elongate so that the fruit can have more physical room to get bigger. So what's going on here is that there's an enzyme, gibberellic acid 3-beta hydroxylase, which converts GA20 to GA1. So the dwarf peas that you worked with in lab, there was just a 1 base change in the DNA for that enzyme. And what results in that is that the enzyme is only 1/20th as active compared to the non-mutated enzyme. Okay, so there's just that 1 base change in the DNA that codes for that enzyme. So the dwarf plants still have all the necessary receptors for gibberellic acid. It's just the enzyme that is a little bit tweaked there, and so it has a very low rate. So they still respond to gibberellic acid. That's not, they have all the receptors and all of that. It's just that they don't make their own gibberellic acid to the level that's needed. And so they stay in the dwarf phenotype until, if you spray them, then they will respond to that. because they have all the necessary signal transduction mechanisms in place. They just don't make much of the GA1 themselves. Okay, we'll talk a little bit more about gibberellic acids. All these hormones, there's a separate slide set that we'll get into on how we have been making use of these hormones in primarily in commercial production of plants. We'll look at that. Ethylene is one of the gaseous hormones. So we talk about the classic 5 hormones. So auxins, cytokinins, gibberellins, ethylene, and abscisic acid are the classic 5. Now there's been more discovered since then, of course, but ethylene was the first gaseous one to be discovered. And so you're looking at it, it's just ethylene. It's not like there's a bunch of different ethylenes out there. It's just ethylene. And so this hormone is found in, of course, plants, but then it's associated, bacteria and fungi can also make it. So you played around with that a little bit in lab with the cut apple, and did we use tomatoes, I think, right? And then I actually set that up for you, where we then put the cut apple and then we bagged it so that the gaseous hormone would stay around the plant. And it should have shown the epinasty, where the branches and leaves all kind of drooped. It should have looked like it was wilted, but it wasn't wilting. It was just this epinasty growth response in when it's exposed to too much ethylene like that. So this is a complicated pathway. The only thing the part that I want you to know is right here, this offshoot. So you go from 8-O-met S-adenosylmethionine to ACC. You need this ACC synthase enzyme. So 8-O-met to ACC, you can abbreviate it. You need the ACC synthase. And then to go from ACC to ethylene, you need ACC oxidase. That pathway, that little part of it, I want you to know and think about. ACC oxidase, you see oxygen is needed. Okay, oxygen is needed to get this to go all the way to ethylene. In the absence of oxygen, you won't get ethylene. I'll come back to that idea. That's important. You also see in the kind of I guess it's grayish blue and the pink boxes, you have a variety of things that seem to be either stimulating or stopping. So when you see this line with a short little line at 90 degrees, that means it's stopping it. So it's saying that you can get inhibition of ethylene synthesis. So these chemicals, I'll come back to these abbreviations. These are chemicals that they've discovered you can expose the plants to, and it messes with this enzyme, and so they won't make ethylene. On the other hand, you can see that there's a variety of things that can stimulate this enzyme and push this pathway, usually all the way to ethylene. So you can see here, indolacetic acid promotes ethylene. So auxins can stimulate ethylene production. We'll get into some of this fruit ripening and flower senescence when we look at some of the applied uses. And then actually when we get into abiotic stress, we'll circle back to ethylene, because that's often involved. So you can see here, wounding, chilling, drought, flooding. These are all abiotic stresses that will stimulate the production of ethylene. Okay, so we'll work through some of this in greater detail. And so you see here anaerobiosis. I never really phrase it that way. In an anaerobic environment, you can get this enzyme greatly shut down. All right, so anaerobic just means without oxygen. Anytime you see A or AN on the front of a word in science, that means without. Temperatures greater than 35 also. Okay, so we'll get into some of that. So this is really what I want you to know, this part. Okay. And as we continue on with the applied uses of ethylene and we get into the abiotic stresses, I'll circle back to this idea and the synthesis of ethylene. So we know for sure, as that some of what was on that diagram, that you can stimulate ethylene production by the plant by other hormones. And then what's being shown here is that epinastic response, where you have the leaves and the petioles and any branches that would be drooping in the presence of too much ethylene. It's kind of odd. I don't know if you've ever gone by a field of herbaceous crop plants after a flood, and it's weird because you'll see them doing an epinastic response. You'll look at it and you might immediately assume that they're wilting. But then if you learn that there's been a flooded, the field was flooded for a period of time due to heavy rain, it doesn't make sense. You're like, well, wait a minute, they just had plenty of rain. Why are they wilting? And it's not a wilting response. It's epinasty, and that's not wilting. And so epinastic, that's an actual growth response, asymmetrical growth that's causing that bending down. And what I won't go into it right now in any detail other than to say, when a field gets flooded, a crop field gets flooded, a lot's going on at the microbial level in that flooding event that creates certain chemicals and gases that stresses the plant, and then they kick into ethylene production, and then they exhibit epinasty. And so it's kind of this domino effect due to prolonged flooding, which leads to prolonged anaerobic environment in the soil. And then the anaerobic bacteria kick into high gear, and they start producing all kinds of interesting compounds and toxins that the plant can't deal with very well. Physiological effects. Fruit ripening, exposing fruit to ethylene, some fruits, causes them to ripen up much quicker. I do that a lot if you go to the store and get a bunch of green bananas, that's your only option. If you cut an apple and put it into a bag of some sort and close it down with the bananas inside as well, you can get your bananas to turn yellow much, much quicker. And so bananas are famous for responding to ethylene. They'll go from green to yellow, ripen right up in the presence of. And so what you're doing is when you cut the apple, you're wounding the apple. And when you wound the apple pumps out a bunch of ethylene. Here you're looking at root hair formation. On the left was exposed, and on the right is more typical. And so exposing the roots to ethylene. And so the plant is capable of doing this, but you can also do it exogenously, where you directly expose the plant to it through your actions. So this one's there's some important exceptions to this. It suppresses or delays flowering of many important crop plants. Ethylene will do that, except it actually stimulates pineapple flowers. So that's a notable exception. If any of you have gone to Hawaii, I've had good fortune to go there, where they have a lot of pineapple plantations. They will actually release ethylene into those fields in order to try to promote consistent flowering across the whole acreage. And so you can do that without gassing. Like, they don't have to put a huge tent over the field and gas it. They don't do that. They actually have a liquid chemical that they add into the irrigation, and it's called Ethafon. And when you just add that into the irrigating water, the plant will take that chemical up and quickly convert it to ethylene once it's brought up into the plant body. So it's pretty easy to do, and that way it can kind of synchronize all the flowering, hopefully, so that you can have an easier harvest once you get the beautiful pineapple. It also, ethylene can stimulate the final stages of leaf abscission. It's actually a multi-hormone impact here, in the sense of some hormones have to go away and then ethylene kicks in. And so in a normal leaf, during the height of summer, what you tend to see is that auxin is being made in those leaves, and often cytokinins as well. That's interesting, they didn't draw that in there. But anyways, what they're trying to show is that auxin is bathing the petiole of the leaf, okay, the leaf stalk. And so the petiole is getting plenty of exposure of auxins and cytokinins. And that's telling the plant this leaf is good, it's healthy, it's doing its thing, leave it alone. Then as fall rolls around, like now, what they're trying to show here is that auxin production plummets, and so does cytokinin. And so you get those down-regulated in the leaf. That's the signal to the plant to start siphoning off all of the useful nutrients in that leaf tissue and send it down to the roots for winter storage. And so it makes sense that if you're going to amputate part of your body, you take all the useful stuff out of it first before you do that. And so that whole process has been going on that we've had the good fortune to enjoy here during fall. And then you see ethylene production ramps up at the last bit of it, so that you can get that weakening. So there's an abscission zone. It's just a thin zone of cells that run in cross section to the petiole. And all of those cells start to weaken. That ethylene bathes those cells. Those cells start to, their cell wall starts to break down, and they just start to degrade. And it's in this zone-like area of the petiole. And then that causes the leaf to fall off. So it's definitely a complicated procedure, but it makes sense that it's complicated because you don't want the leaves to just fall off while they're still green. There has to be a lot of signaling going on to prepare and make the plant get all those nutrients out first. Okay, so we'll talk more about that hormone in more detail too, but wanted to introduce the abscisic acid. Again, that's just one single compound. And this one was, I believe, discovered before ethylene, and it is basically everywhere. Everywhere they've looked in the plant, they can find it being made in those parts. And so basically, I kind of laugh because they usually phrase it like this. Abscisic acid is synthesized in cells containing chloroplasts or amyloplasts. Well, that's pretty much any plant cell. You know, certainly chloroplasts, you're thinking about leaves and some stem tissue. Amyloplasts are often in every single cell, but in particular in roots, root tissue, root cells have a lot of amyloplasts. Oh, yeah, pretty much everywhere. And unfortunately, it was termed the stress hormone before ethylene was fully studied. Really, ethylene should be the stress hormone, but you'll often hear people say, or they'll print it out in a textbook, that abscisic acid is the stress hormone. I would say yes, but in limitation compared to ethylene. Ethylene is way more a broad array of different stresses will trigger ethylene. The reason they call it the stress hormone, abscisic acid, is because they were doing a lot of work with drought and how drought impacts plants. And they discovered that abscisic acid really gets ramped up when the plant is experiencing drought. So here you're looking at the leaves and those cute little guard cells that are surrounding the pore. So these are stomata or stoma that you're looking at. And on the left, it's wide open. That pore is pretty much wide open. And on the right, the guard cells have relaxed. Water has rushed out and they've relaxed and started to close over the pore. So they noted this and they started asking what is controlling that ability of the guard cells to respond under drought. And so what they discovered was that as the plant is assessing that the soil is drying out, it's the roots, right? The roots are picking up on that. Uh-oh, I'm surrounded by a bunch of soil that's drying out. And what tends to happen then is in the roots, abscisic acid gets made very quickly, and then it courses through the plant body and gets all the way to the leaves very quickly, and usually through the xylem. This is all happening. And then it goes to the guard cells. Abscisic acid actually will go to the guard cells and dock on receptors there and tell the guard cells it's time to close over. So it's again, it makes sense when you think about it that it's actually the roots that are the area that is massively producing abscisic acid in response to that drought that they're experiencing. And so, of course, the benefit to all that, to this whole process, is that if you can close down your stomata, you're able to conserve water, and hopefully you won't desiccate and die during that drought. So they discovered abscisic acid concentrations greatly increasing in the root tissue, and so they said, oh, it's the stress hormone to water stress. But ethylene is actually responds to a bunch more stresses. This is a drawing that Laura made. Laura was a honors student in this class years ago, and part of her project was to draw. She was an artist, art and design major, or double major, minor. Anyways, so she did a nice job drawing this out, showing the flow, how the root is assessing and sensing, and then synthesizing the abscisic acid, so it got into the leaf, and making it all the way to the guard cells. And she's trying to show the physiology here, how you'll get, in order for the guard cells to close, they have to lose water, so there has to be a condition where water potential shifts, and you get water moving out of the guard cells into other areas that are more negative nearby. So here's the abscisic acid docking in the receptor to kick all that off. So it's complicated. This is from the textbook showing kind of the overview of that. For this class, the major upshot is that you have the abscisic acid making it to the guard cells, docking in its receptor there, and causing water loss, essentially through water potential laws, to get the water to flow out into less negative to more negative. So you can graph this data out. Here's showing on the x-axis, you have time in days, and then you have a couple of things going on here. The top graph is showing water potential in the leaves. And so you can see that once the dotted line is stop watering, and you can see that the water potential gets more negative as the leaf cells are under stress, water stress. And then what you see in response is that the stomatal resistance goes up. That just means for resistance to go up, the pore must be getting smaller to increase the frictional effects. And so the friction of the water vapor molecules creates more resistance. So stomatal resistance means the pore goes smaller with greater resistance. So the pores are shutting down. And then you can also see a very nice mirror of that with the abscisic acid synthesis. And so it kind of goes hand in hand. And then once they water the plant again, you can see a reversal of how the water potential starts to become less negative in the leaf. And as a result, the stomata open up and the abscisic acid goes way down. So really nice data to support this idea that you have abscisic acid playing a really crucial role in closing down the stomata under water stress. Okay, so those are the so-called classic 5 hormones, but now they've discovered others, and I'm sure they'll continue. So this is actually similar to some animal steroid hormones that we have, the racinosteroids. And so if any of you have taken animal phys or human phys, that may look vaguely familiar. They're still working out the details of this group of hormones. I'll just say a few things right now, and I'll mention a few later on. But right now, it seems like it's really important in lots of different things that the plant has to deal with. Cell division and elongation, so that's growth in general, plant growth. Germination of seeds, senescence is just the natural dying process that they have to go into during certain stages of their life, the last stage, and then different stress responses. So they're finding that it's very heavily involved. This picture is looking at mutants of maize. So I think this is looking at impacting sex determination in the plant. This is corn. And so that's a pretty important impact if it can mess so strongly with the sex of the plant. And so the wild type is on the left. This is the male plant with the so-called tassel that has the pollen. And then you can see here on the right, this is feminized, they say. You can see the kind of like the corn on the cob idea starting to establish here. And so it's definitely very different from the wild type. And so some very profound impacts on this stage of the plant's life cycle. So a pretty big deal. So they're pretty intensely looking at that in particular, because it certainly plays a heavy, heavy role in plant development. Okay. So, and then some others that they've recently discovered, salicylic acid, which may sound vaguely familiar to some of you that have some chemistry background, because it's related to acetylsalicylic acid, which is hard to say, which is aspirin, which they've discovered initially from the bark of willow trees. So the Native Americans knew about that way back when and would use the bark from willow trees as a source of pain reliever and anti-inflammatories. So salicylic acid is a plant hormone. They're using it as a hormone. Jasmonic acid is another one. And so we will certainly touch again on these two because they're very heavily involved in plants defenses when they're getting attacked by a pathogen or an herbivore of some sort. So we'll mention them in context to that in a few weeks. And then also abiotic stress. They're very important in serving as part of the signal transduction during both of these. So I will definitely come back to these and say more about them. And then probably strigolactones of the newer hormones. These were probably the first to be discovered even before the steroids, I think. And strigolactones, the reason they're called that is that they are named because of their ability initially to stimulate the seeds of the striga plant or striga plant. So striga is a parasitic plant. And what tends to happen is if it gets into a conventional, crop field, it can cause a massive amount of issues. Especially, I've read mostly about corn acreage that's in production to corn. If the striga plant germinates and grows in those fields, what it does is it plugs into the corn plant and starts siphoning nutrients off of the corn plant. So the growers then are left with corn plants that are struggling. The corn on the cob production can plummet. So here you're seeing the base of a corn plant here, the stalk of the corn plant, and then all this really pretty plant, the beautiful red flowers, that's striga. And so what it's doing is on the right, that bigger part of the image, that's the corn stem. And here is the stem tissue and root tissue. I think it's more the root tissue that they call it a haustoria. That's kind of a general term. Someone says, oh, look at the haustoria. What they're implying is there's some kind of tissue that's invading another tissue, and they call it a haustoria. So it's just trying to get at the idea that the roots of the striga plant are plugging in to the corn and siphoning off the nutrients. So this has been a problem for decades, and they've been trying to figure out how to control striga. And then they realized that the plant, all kinds of plants produce strigolactone or strigolactone. And unfortunately, it can stimulate the seeds in the soil. When the seeds get exposed to it, that'll start causing them to germinate and grow. So they're heavily looking at that. They're trying to find ways to control this parasitic plant through genetic engineering and ways to use biocontrol. Everything that I've read about so far has met with pretty limited success. So they're still working on it. Okay. So we need to think about all these hormones. They're very important for the signal transduction idea. We need to think about the hormones being synthesized in certain parts, possibly multiple parts of the body, and then they can act locally, they can go distant, and then they will help with the signal transduction wherever they need to send that signal. And so that's kind of what's being shown on this, but they're also showing the idea that once the signal transduction hopefully is successful and the shift in gene expression has occurred and it's kind of reached its conclusion, then you need to shut that down. You can't keep having it running full tilt and keep doing it necessarily. It's context dependent on what the behavior is for the signal. But on the right, you're seeing the idea of, okay, now we got to quiet this down and stop this hormone from acting. And so you could do that by just degrading the hormone chemical, breaking it down. You could conjugate it, often with a sugar, so it stays intact. It's just once it's conjugated, it doesn't stay active. You could stuff it somewhere, maybe in the vacuole, or pump it out. And so it makes sense that there's these checks and balances on the activity and presence of the hormone. So this getting the hormone around the plant body, if you're thinking like a short distance idea, certainly the hormones can go from cell to cell, and that can happen through diffusion properties, that it diffuses through the cell membrane of the cell, or it can happen, it's often more so that there's a protein, a channel protein, or somehow that allowing for the diffusion across. And then, of course, if you need to get the hormone across and against its concentration gradient, often you'll need to expend ATP to get that to happen. So you have to think about that. Sometimes it's a cost to get the hormone delivered. If it's long distance, it makes sense that you can see xylem and phloem transport. And I should have put one more here. I thought I did. Plasmodesmata from cell to cell. So plasmodesmata. You need to edit that. All right, so it just depends on the hormone, whether or not it's what way this will happen. Certainly makes sense that hormones can flow through the xylem and phloem because it's a very efficient way to send. Some of the volatile hormones, on the other hand, they actually get released out of the leaf and can, if you think about giant trees, for example, that have a massive multi-story canopy system to their branches and leaves, it's common that they'll release some of these volatile hormones out, and then that travels just in a gaseous state through the outside and around the leaves and branches. We'll talk more about that later as well. And bringing back this image that I showed you last week about the odd mechanism that auxins have when they have to go from cell to cell, you think they just go right through the plasmodesmata, but they don't seem to do that. It seems to be controlled by proteins that will allow for transport of the hormone in with a proton that comes with it. So in order to get that proton, you've had to expend some ATP to some extent in order to get that. And then you can see that the hormone comes into the middle cell, and what I'm trying to show here, it'll travel and then exit that cell through special transporters again and keep doing that. So very odd, kind of complicated mechanism, but one that they could definitely show. And then I did also want to remind you about this idea with electrical signaling, that this can be a method that plants employ even for long distance signaling. They can do electrical signaling. It can certainly be used in very short distances, like with a Venus flytrap that's actually electrical signaling that's partly causing the trap to close, but it can also be used in longer distance signaling through the plant body. And we'll talk more about this too when we get into how plants defend themselves. And so we're actually, if you've had any kind of human or anatomy or neuroscience class, the idea of an action potential as a way to send that electrical signaling through from cell to cell to cell to cell. And so if you've taken those classes, this may sound familiar. I'm not going to get into this in this class, but for that action potential, you're looking at a transient depolarization of cell membranes, and it's done by ion fluxes. As you go down, keep following the signal, you'll have these ion fluxes due to these voltage gated ion channels. So it's pretty, I always scratch my head when I think about this because it's pretty complicated, and yet how quickly it can work in our brains and in other animals and also in plants. It's pretty quite amazing. So we'll get a little bit more into this as well. Electricity, electrical signals. Okay, so the next set of slides I want to talk to you a little bit about. We're not going to get into the details because it can get quite complex very quickly. But they've been studying how do the hormones do their thing when they get to their site of action. How are they actually getting that transduction to occur, getting the signal to be heard, so to speak, and altering gene expression ultimately. They've actually come to the conclusion from the data they've been gathering that a lot of it's actually being derived from bacterial methods. And so this is a long evolutionary history that they have evolved, from microbes are the first thing on the face of the planet. And so we all derive from that, really. And so thinking about this idea of bacterial two-component signal transduction systems or pathways. And so I'm going to explain the basics of that and then show you how plants have taken that and co-opted it and put their own spin on it. And so, but I'm going to keep it again in a big picture idea because it can get quite complicated. But this one, the basic two-component, what they mean there is that there's two major enzymes needed for a two-component to work. And so this is one of them. It's a sensor kinase protein. And when you hear the word kinase, what that means is that it's a protein that uses phosphorylation to send a signal. So you're going to see a phosphate group involved in this, okay? The other component is the response regulator protein. Okay? So these are the two components. And then the rest of the phrase, signal transduction, that just means that they're heavily involved in getting the signal coming from the environment transduced and amplified to the interior of the cell until you finally get a shift in gene expression. So the idea here is the kinase protein will have a phosphate group that it can then pass on to the response regulator. And that simple passing of the phosphate group from the kinase to the response regulator is enough to tell the response regulator protein to do a response. All right, so it's a nice simple signal mechanism that can happen quite quickly. So this is from your book. So the very left figure A is looking at how bacteria do it. And so here's your sensor protein kinase up top. You don't need to know all the nitty gritty, just that it's a sensor protein kinase. And you can see the phosphate group in the circle here. So there's the phosphate group. And they're showing it being transferred to the response regulator. So that event, then I told this in my other class too. We just covered two components in my other class too. But basically, I think of it like a relay race where the phosphate group is like a baton that you hand to the next runner in the race. And then that gives a signal to the runner. Once it's got the baton, it can take off and run, right? The runner can do the race. So that's how I think about it in my head. Once this response regulator gets the baton phosphate, it can take off and do its job, which is to alter gene expression. So it will go to the DNA now that it has that baton phosphate and do its job. All right, so very simple. That's a pretty simple mechanism. And yet it allows the signal transduction to happen very quickly and efficiently. So now figure B is how you take that and kind of put your own spin on it. So here you're seeing a so-called middle player in the game of transfer of the phosphate. So really that's, when you look at it, that's the big picture idea, is that you've added in another layer of another person in the race that's going to get the baton. All right, so here you see the kinase delivering it to this histidine phosphotransfer protein. All right, and so that's the middle player, the second in line in the race, and then that one delivers it to the response regulator. So you can see it's a little bit more sophisticated or complicated, but it's the same basic idea, passing the phosphate baton from one protein to the next until the response regulator does get it, and then that can go and interact with DNA and shift gene expression. Okay, so then the next slides are looking at a variety of ways that plants kind of take some of these signaling pathways and are operating their signal. Now, I rarely say this to you. Don't look at these slides until I talk to you about them. All right, I'm going to ask you not to even spend time on these until I talk to you about them. Because if you try to figure these out, at least I think if you try to figure these out on your own, it's just going to be an exercise in frustration, and you'll probably overstudy. There's a few key things I want you to get out of this, and then we're going to call it quits on it. Okay, so hold off, and we'll let's see. I guess we'll do it on Wednesday. We'll go over this on Wednesday.